all of you to our American Thoracic Society RCMB Lung Aging Research Group webinar series, and myself as a chair, Irfan Rahman. We have my colleagues here, the co-chairs, Benjamin Singer, Marika Lehman, Beata Kosmedar. So as you know, the lung aging group, we have our main goal is to increase the awareness, disseminate knowledge of pathophysiological mechanisms and interventions, repair of aging in the lung, and interest in terms of basic biology of aging, lung aging in debilitating lung diseases. So this been our goal, and we started about 10 years ago, and then there were some interruptions during pandemic, but we continued the mission. And we have a really good group here, and we have, you can go online and take a look our assemblies and RCMB working group, especially the lung aging group. And we would like to welcome all. And this is a group where we strengthen ties with other societies. For example, I was the president of inhalation of the Society of Toxicology. So we bring that, the FASEP group, then the other free radical societies. So this is also we collaborate with NIH institutions. We'll have a session going on in the next 8 years. And please do join us with the NIA and NHLBI collaborations, and then increasing and supporting our memberships. This is our one of the main goals, and we would love to you to come up and tell us what we shall do. And of course, the networking for education and research for lung aging and critical and other lung diseases. So Ben, you'd like to talk about some missions and mentorship, please?
Sure, because we started a few minutes late, I'll keep this short, but I want to welcome everyone to this webinar. We're so proud of this series, bringing together thought leaders in aging biology to really support the missions that Irfan mentioned. So that really is our core within the RCMB, who has a broader mission of promoting lung biology at the cellular and molecular level within the larger ATS. And our working group focuses on the dimensions of those biologies that change over time. That's the fundamental of aging. So to keep going with the webinar, I'll turn it over to Dr. Kosmeider to introduce our first speaker.
So yeah.
Good afternoon, everyone. My name is Beata Kosmeider from Temple University in Philadelphia. Thank you for joining us today. I would like to introduce the first speaker, Dr. Chang-Fu Yao from Cedars-Sinai Medical Center. His research focuses on lung fibrosis and lung development. He utilizes animal models, human samples, and advanced technologies to define underlying molecular mechanism of lung injury, repair, and progenitor cell function and regulation. And today he will talk about the senescence of alveolar progenitor cells in pulmonary fibrosis. So please go ahead.
So while we are waiting for Dr. Yao, yeah, okay. Please go ahead. Yeah.
Okay, I'll get started. And first of all, I would thank the organizer to give this opportunity for me to share our work. And since this is aging lung interest group, I don't think I really need to introduce what is IPF, but I still want to make this point that IPF is a age-related disease. The average diagnosis age is about 66 years old, and so far there's still no cure. Talking about aging, cellular senescence is a hallmark of aging, and we cannot avoid it. And there are many stimuli can cause cellular senescence, like epigenetic factors, telomere shortening, DNA damage, mitochondrial dysfunction, proteostatic dysfunction, or nutrient dysfunction. It can also lead to proteostatic dysfunction, nutrient dysfunction, or chronic inflammation and stem cell exhaustion. And there are many features of senescent cells. To identify senescent cells, we need to look at probably not not if all of the features, at least not one is not enough. These features including like chromatin reorganization, like increased the senescence associated H chromatin foci, increased gamma H2AX foci, and the cell cycle arrest increased the cell cycle inhibitors like P16, P21, and the increase of RS signaling, mitochondrial dysfunction, pro-survival pathways. And it also shows morphological change like enlarged and flattened cell shape or disrupt nuclear membrane. It also shows increased senescence associated with beta-gal signaling, lipofuscin, or I mean, one of the other important factors is the secretion factors like cytokines, chemokines, MMPs, EVs, and all these factors have been considered as the senescence associated secretory phenotype, so-called the CESP factors. And when we really need to look quant through all those features to identify the senescent cells. And the lung epithelium is composed by multiple epithelial cell types. And at a different region, has been considered that a different progenitor cells will be responsible for local injury repair. And in the airway, we believe that basal cells, which are marked by T5, P63, and NGFR, so as the progenitor cells, whereas in the distal alveolar region, alveolar type 2, the 82 cells, which marked by surfactant proteins like SF-TBC, TBB, TBD, or A2, or and HT280 serve as the local progenitor cells for injury repair. And when we compare the cellular composition of epithelial cells that isolated either from a normal donor distal lung tissue or IPF explant tissue, we found that NGFR labeled basal cells are significantly increased, whereas the HT280 labeled type 2 cells is a significant decrease in IPF explant tissue compared to the normal donor distal lung tissue. And this cellular composition change is not really restricted to IPF, but also applied to other lung fibrotic lung tissue. Here I show example of PASC, post-acute sequela of COVID-19, aka the long COVID sample. We can find that they also shows a decreased percentage of type 2 cells and an increased percentage of basal cells. And the further one, we try to identify what pathway has been changing for the epithelial cell types. We found that the cellular senescent pathway is among the top dysregulated pathway for many epithelial cell types when we compare either PASC or IPF to the controls. And this includes alveolar type 2 cells and the transitional cells, which I'm gonna talk about more in the later of the talk. And further, if we send senescence associated beta-gal on IPF explant tissue compared to the donor tissue, we found that there is a significant increase of senescence associated beta-gal signaling in multiple cell types in IPF explant tissue. This includes the potential epithelial layer here. And GWAS study have linked genetic mutations for IPF to host defense defect and cellular senescence, as well as gene mutations that involve in regulated type 2 cell functions. So we actually made a bold hypothesis that what if the senescence of type 2 cells may possible drive the progressive lung fibrosis. First thing we want to validate whether there is type 2 cell senescence in human IPF. So in the single cell RNA-seq data, we compared the IPF type 2 cells with donor type 2 cells. So we observe a significant increase of cellular senescence activation score. At the same time, we also identify senescence pathway and senescence associated pathway are significantly upregulated in IPF type 2 cells, donor type 2 cells. And when we co-stain type 2 cell marker SF-TBC with the cell cycle inhibitors P16 or P21, we found that the double positive of P16 and the pro-FTBC or P21 with pro-FTBC are significantly upregulated in IPF compared to the donor distal tissue. And this indicates that the possibility that the senescence of type 2 cells may contribute to the general pool of senescent cells in IPF. So next, we want to validate whether actually type 2 cells senescence can drive lung fibrosis. So we made a mouse model to test this hypothesis. And the gene involved in this model is called sensory A. Generally, people believe sensory A serves as a scaffold for HDAC to interact with the histone, then de-escalate the histone involved in gene repression. But at the same time, sensory A can also interact with non-histone proteins like p53, which we're going to talk more in the later of the talk. And in our developmental study, when we use sonic hydroxyurea to knock out sensory A from developmental endoderm, we observe a lung-specific defects. And what's more, we found that the epithelial progenitor cells shows increase of senescence associated beta-gal signaling and increased gamma H2X signaling. What's more, we found that most of the epithelial cells have been arrested at the G1S transition, which met the criteria or the definition for senescence is that the cell has been permanently inhibited in the cell cycle. So this altogether indicates that this epithelial progenitor cells has been, or they have acquired a senescent-like phenotype. So we want to test whether the adult type 2 cells will also acquire the senescent-like phenotype if we knock out sensory A. So we use SFTP-CQER to knock out sensory A in adult type 2 cells. Here we call the sensory loss function. Basically, we gave 200 milligrams per kilo tamoxifen every other day to 10 to 12 weeks old young mice. And we found that basically around 2 weeks, it takes about 2 weeks to achieve sufficient knockout of sensory A. Then we realized we actually found that the cell size of the sensory loss function type 2 cells keep increase along the time. And by the time of 6 weeks, the average size of the type 2 cells which lost sensory A has been increased about 3-fold compared to the control type 2 cells. And when we perform single-cell RNA-seq analysis, we found that the sensory loss function type 2 cells also shows increase of cellular senescence score and also shows upregulate basically the same pathway we actually identified when we compare IPF type 2 cells with donor type 2 cells that are senescing and senescing associated pathways. We also performed a 6-week long IDU water labeling, and we found that the sensory loss function type 2 cells, basically there's no IDU signaling has been detected, which means that during the 6-week time course, we observed there's no sensory loss function type 2 cells have ever entered a cell cycle, which means that they lost their capability of cell cycling. This also meets the criteria of cellular senescence. And when we perform as senescence associated beta-gal staining, we also observe increase of senescence associated beta-gal staining in the sensory loss function mouse lungs. So all this data together indicate a possibility is that the adult type 2 cells, when they lost sensory A, they also acquire a senescent-like phenotype. Then what happened to the mouse lung and the mouse itself? We found that the sensory loss function mice have shows a body weight drop and also get about 30% for mortality over a time period of 8 weeks. And furthermore, we found that it shows tissue or lung tissue consolidation start around 4 weeks, and usually at the distal end of the lung, and further progress along the peripheral. So this is the mesenchymal staining. You can see the increase of the blue, which is the collagen staining at the tissue consolidation region. And this was actually really resemble what happened to IPF adaptation that the lung fibrosis start from the peripheral towards the inside. And furthermore, if we stent for alpha muscle actin, we can observe a significant increase of the alpha SMA signaling in the tissue consolidation region. And this was also confirmed by Western blot. So this indicates this increase of myofibroblasts in the tissue consolidation region. And we also observed a significant increase of hydroxyproline content. So this indicates that the mouse lung loss of sensory A really developed spontaneous and progressive lung fibrosis. Next, we're going to touch bases on some of the pathways we are interested. And here I'm only going to show p53 and T-beta. And the reason why I'm going to talk about these two pathways is just because when we perform upstream analysis using different data sets, which here I include background seq of isolated AT280 cells from donor or IPF tissue, or single cell RNA-seq compare IPF type 2 with the donor type 2. Background seq of the 6-week time point sensory loss function with control, or single cell RNA-seq compare sensory loss function type 2 with control type 2. That TGF-beta1 and p53 are always among the top upstream regulators we identified. And further, we found that the sensory loss function type 2 cells really shows a significant increase of p53 activation score, which is the same when we compare IPF type 2 with donor type 2 cells. And when we perform IF staining for p21, we found that the p21 can only be detected in the sensory loss function type 2 cells, and also shows the increased transcript level in the single cell RNA-seq data. And p21 is a downstream activation marker for p53. So this all indicated there is activation of p53 in sensory A loss function type 2 cells. And what's more is we actually observe a increase of p53 protein level. So when people talking about the activation of p53, we're looking into the phosphorylation of p53. Here, what's different is we actually observe is a stabilization of p53 protein. This was not actually restricted to the sensory loss function model. We also observed this in the bleomycin injured mouse model. And also, when we compare IPF type 2 cells with donor type 2 cells, so we also increase a stabilization of p53. And so what if we knock out p53 from the sensory loss function mice? We found that the double knockout shows partially rescue of the body weight drop and also rescue of mortality. It also shows partial rescue of the fibrosis phenotype, and also shows partially reduce of senescence-associated beta-glial signaling. And since, as I mentioned earlier, we observed this stabilization of p53, and we know that the p53 degradation is regulated by E3 ubiquitinase MDM2, mediate the p53 ubiquitination for downstream lysosome degradation. And what if we now have MDM2 in type 2 cell to achieve the p53 stabilization? We generate this SFTP3-ER MDM2 loss function mice, and we found that just by a single dose of 100 milligram per kilo tamoxifen treatment, by 4 weeks, we already start to observe lung fibrosis. So this indicates that stabilized p53 in type 2 cells can lead to lung fibrosis. And further, people also show that the escalation of p53 shared the same lysine residue with ubiquitination. If further mutate the C-term lysine residue to exclusion mimic, it can prevent MDM2 mediate p53 ubiquitination. So we want to see whether there is increased escalation in the sensory loss function type 2 cells. As I show here, you can see that there's increased level of p53 escalation in the sensory loss function type 2 cells compared to the donor type 2 cells. And further, we also found that the type 2 cells from bleomycin injured mouse lung also shows increased p53 escalation level. And the literature shows that the major p53 deacetylase is HDAC1 or the SEARCH1. And when we compare the LGRC microarray data set, we found that HDAC1 expression level doesn't show significant change, but SEARCH1 shows a significant decrease when we compare ILD with control patient sample. So we hypothesized, or we tried to test whether there's a potential that the SEARCH1 will serve as a major HDAC acetylase in type 2 cells. So we performed some in vitro assay using a mouse type 2 like cell line. Here we use MLE15. And as here I showed that we can use, when we treat MLE15 with TGF-beta1, it shows decrease of SEARCH1 express level. And at the same time, it induced the escalation of p53. If we active SEARCH1 expression by resveratrol, and it can reduce this increased level of p53 escalation. There's another way we can induce p53 escalation is using doxorubicin to treat the MLE15. Here I also include the SERP1 siRNA. You can see that SERP1 siRNA can downregulate the SERP1 express level and induce some increased level of acetylation of p53. And the doxorubicin can also induce the increase of acetylation of p53. But if we combine them together, it can significantly induce much more strong band here you can see of acetylation of p53. So this data combined indicate there's a possibility that the SERP1 may serve as a p53 deacetylase in type 2 cells, and also may bridge between the p53 and the TGF-beta pathways. Then we want to see whether actually the p53 acetylation can direct in type 2 cells can direct lead to lung fibrosis. So we generated this lentiviral vector which encodes over p53 K2Q mutation at the C terminal to mimic p53 acetylation, which drive by mouse TPC promoter. So when we deliver this lentivirus to the mouse lung, and we found that just by, it doesn't actually need a very long time. By 7 days, we already can observe significant induction of lung fibrosis. So this really indicates that acetylation of p53 in type 2 cells can drive lung fibrosis. So next, I'm gonna move to TGF-beta activation in type 2 cells. And back to 2020, there are a few labs, actually also include us, that identify a cell type, or I should say more like a cell state, that in between of type 2 to type 1 differentiation. And in this cell shows high regulation of senescence signaling, p53 and the TGF-beta. And when we perform ligand receptor interaction for our single cell human single cell RNA-seq data set, and we found that in the control, PDC has been predicted as the major receiver for TGF-beta. But in IPF, the transitional cell has become one of important receiver for TGF-beta signaling. But in the long COVID sample, the transitional cell become the only important receiver for TGF-beta signaling. So this data indicates a possibility that the transitional cell may play an important role for TGF-beta activation in this lung fibrosis disease. So we go back to our single cell RNA-seq data. We can identify the transitional cells in our sensory loss function mice, and these cells are solely derived from the loss sensory loss type 2 cells. And we can also observe this cells showing high TGF-beta signaling p53 score and senescence score. Same thing when we compare IPF sample with donor sample, the IPF transitional cells also shows high senescence score, p53 score, and TGF-beta score. So we also perform single cell RNA-seq analysis on bleomycin injured young mice and old mice. And we know that the a single dose bleomycin injury for the young mice as a resolving lung fibrosis model, where the old single bleomycin injury to the older mice is related to a persistent lung fibrosis model. And in this data set, what we found is that the transitional cell ratio compared to the type 2 cell ratio for the young mice, they first increase, then decrease very dramatically. Whereas the older mice, it shows relative maintain the high percentage of the transitional cells versus the type 2 cell percentage, as I show here. So this really indicates a possibility is that this transitional cells may promote the persistence of the lung fibrosis in the old mice model. So we go back to our sensory A loss function mouse lung, try to identify where this transitional cell locate. So with Stanford Claudin 4 creatine 8 and the GFP, we found that the Claudin 4 high, creatine 8 high, and the GFP positive cells only locate at the peripheral of the mouse lung and near the fibrotic foci. So one thing I try to just emphasize is that I want to use the creatine 8 high as the marker just because actually creatine 8 is expressed by all epithelial cells at a low level for the alveolar cells, but high level in the airway. And this in the alveolar, only the high ones are more labeling the transitional cells. So just one thing I want to emphasize. And we also found that these transitional cells are high for ITGB6. And we know that ITGB6 is the activation marker of TGF-beta1. So we further check TGF-beta activation marker phosphoSMAD2. We found that phosphoSMAD2 is positive in this transitional cells and the surrounding stromal cells, which indicate there is a general TGF-beta activation at this region. So what if we block TGF-beta activation by taking out TGF-beta receptor 2 from the sensory A loss function mice? So we found that the double knockout can also rescue mortality and the body weight drop, also shows partial rescue of the lung fibrosis. And what we didn't expect, it also reduced the senescing associated beta growth signaling. And when we further looking at whether we still can identify those transitional cells with Stanford Claudin 4, creatine 8, and GFP, now at this time, we can only find those low-level expression of creatine 8 cells. We couldn't find any those surprisingly high creatine 8 transitional cells anymore. So which basically, they are similar as the control sample. So this really indicates that if we block TGF-beta signaling from type 2 cells, it will not only reduce the lung fibrosis, it also shows this prevention of the differentiation from type 2 cells to the transitional cells. So lastly, we want just try to compare whether it's the senescence of type 2 cells or the loss of type 2 cells gonna driven this progressive lung fibrosis. So in order to achieve the type 2 cell ablation, so we generated this mouse model by SFTC CRIAR close to ROSA DTA allele. For this mouse line, we couldn't do our traditional 3-dose tamoxifen treatment because all the mice will die out in 3 days. So we performed this 1 dose per week treatment, then harvest at the different time point. And but this treatment strategy actually it really didn't show any mortality for DPA mice. The sensory loss function mice still shows progressive lung fibrosis, but the DPA mice that shows some fibrosis, but we didn't observe this progression of lung fibrosis for this model. So this indicated probably is more the senescence going to be more important to driving this progressive form of lung fibrosis. And further, we want to check what if we remove the senescent type 2 cells. Here we apply the senolytic cocktail, the dextrin with quercetin combination. So this also can partially rescue mortality and body weight drop, also partially rescue lung fibrosis, and also reduce of senescence-associated beta-gal signaling. So this data together indicate that it is the SIG or the senescence of type 2 cells is more important driving this progressive form of lung fibrosis rather than just simple loss of type 2 cells. So we come out to this working model is that the TGF-beta signaling, which can cause downregulation of SIRT1 leads to accumulation of acetylation SIRT1, which is the acetylation SIRT1, acetylation p53, and the acetylation of p53 will prevent ubiquitination of p53, promote stabilization of p53, and finally lead to the senescence of type 2 cells and the differentiation to the transitional cells, which lead to the progressive form of lung fibrosis. And at the end, I would like to thank my mentor, lab member, collaborators, and the funding source, and thank you for your attention. I'm ready for questions.
Thank you. It was a great talk. Are there any questions?
I'll start with one, Beta.
I think there is a question in the chat box as well.
Yeah, you can go ahead, Scott.
Okay, yeah. So thanks for a great talk. This is a really interesting work. I'm wondering about our lab, and then also Dean Shepard, Mike Beers in the surfactant protein C group model, and David Schwartz in the MUC5B model have all shown that these transitional cells accumulate when there's activation of some of the proteostasis stress response pathways, the integrated stress response or the UPR. And I'm just wondering if you've looked in your model to see if your loss of SYN3 is actually doing that, if it's activating those pathways in your model.
I actually didn't really see a strong UPR activation in this model. But I mean, I think that's highly possible because the accumulation of p53 may actually stress of ER. There is a report, but I didn't really check UPR specifically.
Sure. Yeah.
And
then,
we have.
And then go ahead. I'll let someone else ask.
Please go ahead.
Yeah, another question gets to the sort of the definition of senescence in these transitional cells in particular, right? The beautiful genetic experiments that causally show senescent cells are linked to important aging phenotypes were done by the late now Judy Campisi, unfortunately. And by Jim Kirkland and Dr. Anderson at the Mayo Clinic. And in those studies, they actually induced, made cells that were expressing P16 susceptible to an apoptosis stimulator, right? And those are very nice genetic data. And that's why the field has relied very heavily on P16 for the definition of senescence, because that's where the causal genetics are. But as you alluded to in your first slide, sort of the definition of a senescent cell has expanded quite a bit to include a lot of biology. The question is are these cells really senescent? And I ask that question because we know from lineage tracing studies, right, that these AT2s are on their way to AT1s, and that a transitional cell can become a type 1 cell, and that doesn't fit my definition of replicative senescence. So I'm just interested in your thoughts on
that. Actually, that's a excellent, really, really excellent question, because if just an increase of P16, other definition could be just cell cycle arrest, but cell cycle arrest probably not a definition of senescing itself, because senescing cell, yes, is arrest for cell cycle, but post-mitotic cells are also arrest in cell cycle. So I don't think P16 can be used as a solely marker for senescence. So that's why in the world of senescing, I think in sorry, I don't remember the year, but Cell published a guideline to identify senescent cells. You really need to crunch through all the features of senescence features to identify whether it is a senescent cell or not. And even in that case, I'm still not confident to call the cells I'm looking at as a senescent cell. So that's why I call it a senescent-like phenotype. And same thing as you mentioned about the transitional from type 2 to type 1, the transition cell actually just fit in between. Whether it can be called as senescent or they just a state increase those senescent-related gene expression since the transition cells is high for for those cell cycle arrest markers, but doesn't necessarily mean they are actually just senescent cells. If they actually can bypass the arrest stage, stop secrete, all the factors that potentially gonna affect the local microenvironment, then transition to type 1 cells, then the kind of exit the more pathological like state, then I don't really think it is a definition for senescent cell for transition cells. But they actually, I think more correct is they showing those features and the has their secretion phenotypes may affect the local environment, which cause more effect on driving the fibrosis. But if some of the pathway, if they can be persistently inhibited in that state, that may be the kind of like a senescent cell. But if just enter that state, and then if the local environment allow, then it can bypass that stage, get into type 1, then they are not senescent anymore. So I totally agree with you. If just a normal condition of, or somehow they can bypass that stage, they doesn't fit of senescent cells definition.
Thank you so much. So I think we need to move to our next speaker, and there are questions in the chat box. Could you please, Dr. Yao, maybe address them this way?
Okay, yeah, sure.
Thank you so much.
Great. Thank you, Dr. Yao, and thank you for the conversation. Our next speaker is Dr. Christina Brandenberger from the Institute of Functional Anatomy at Charité Medical University of Berlin in Germany. She has a longstanding interest in immune activation over the life course, particularly within the context of lung injury and pneumonia. So Dr. Brandenberger, if you can share your screen, we're looking forward to your talk.
Thank you very much. So 1 second. All right. Can you see the screen? Okay, great. So thank you very much. It's a pleasure to be here in this webinar today. And thank you very much for the invitation. And yes, today I will be talking about how inflammation is affecting acute lung injury and pneumonia in old age. And I mean, I'm sure you're all aware about the hallmarks of aging. This has been an updated version of the Lopez-Otin et al. Publication from 2013. And some new hallmarks have been integrated here, including as well chronic inflammation. And chronic inflammation is kind of like low-grade inflammation that is constantly occurring, and mostly including pro-inflammatory cytokines such as interleukin-6, interleukin-1 beta, or TNF-alpha, just as a few examples. And it could be driven by all the other hallmarks of aging, such as, for example, cell senescence as well, mitochondrial dysfunction, impaired autophagy, changes in proteostasis, and so on. And these factors can lead to an increase in inflammatory cytokines, but also other factors like metabolic changes, hormones, or danger-associated molecular pattern, or pathogen-associated molecular pattern activation can also lead to NF-kappa B signaling and increase in inflammatory cytokines. And these changes then have been recognized as inflammaging. And in acute lung injury or acute respiratory distress syndrome, we find severe inflammation in the alveolar region, where we have leukocyte recruitment, we have a damage of the air-blood barrier, edema formation, and this goes along then with hypoxemia and in the worst case, respiratory failure. And I'm sure that everyone is aware now since COVID-19 that the elderly have a much worse prognosis for survival, and they also show a slower recovery rate. But this is not only true for COVID-19, it's also true for bacterial pneumonia and also for other viral pneumonias. However, the reason why is still not really understood. And one factor that we are interested in is how actually inflammation is affecting acute lung injury and pneumonia in old age. And we have been addressing these questions with different mouse models, and I'm going to introduce you some of these studies we have done. Like one with the mouse models of LPS-induced acute lung injury with age and senescence. We have also addressed the impact of gut microbiome and its metabolites in inflammation and acute lung injury, but also mouse models of infection with Klebsiella pneumonia in aging, in particular interventions that we did, as well as in precision cut lung slices and neutrophils to study the infection with the Pseudomonas aeruginosa infection in older age. So I will start with the first one. And here we just started with young and old mice, like 6 mice that we received from Janvier Labs, as well as a mouse model with inducible Cre-Lox-P mouse model under the SFTPC promoter that knocks out TERF1, which is a protein of the sheltering complex, a sheltering complex of the telomeric ends that protects the telomeres, and with its knockout, we can cause cell cycle arrest and telomere distortion, particularly in 82 cells. And we used dosing LPS of different doses, and we looked into different time points, addressing histopathology in survival, inflammation, also alveolar macrophage activation, and we were of course also interested in alveolar type 2 cells with senescence and surfactant metabolism. So when we did the study with the 10 days and we gave LPS to both young and old mice, we had actually only 40% survival in the old animals. And the animals that did survive, their lung histopathology looked quite good. So this is a young survivor, this is an old survivor. So it seems that the recovery took place by 10 days. This is, however, the lung of an animal that didn't make it. And you can see here we have huge infiltration with inflammatory cells and edema formation. So this animal did not make it. So this is actually also to some extent reflecting what we observe in the elderly population with acute lung injury and pneumonia, that they have a higher mortality rate. Now, when we looked into the earlier time points, we could already see at 24 hours that the increased inflammation manifested here in the lung tissue, and then currently progressing, but also in the BALF cells with macrophages and neutrophils. We see here an increased recruitment of neutrophils, which is then further progressing after 72 hours. And this also holds true for inflammatory cytokines in the BALF. We also had a closer look at the macrophages and their activation, and we did actually see that when we looked into surface markers, CD80 and CD68, that are two M1 inflammatory markers, so for the pro-inflammatory macrophage phenotype, we could see that we had a stronger expression in old macrophages with the infection, with the inflammation. And also when we isolated the macrophages from young and old mice and stimulated them in vitro, we could see that the macrophages from old mice that they show more expression of M1 markers, and when we added LPS, this was even accelerated. So also in vitro, without the further environment of the lungs, the alveolar macrophages were more prone to a pro-inflammatory stimulus and response. Also in the lung tissue of the control animals where we did not have any LPS exposure, we could see that we have an increased TNF-alpha response per se in the BALF, and also interleukin-6 in the serum. So there were some signs of inflammation in the tissue, but also in the macrophages with aging, and this also went along with the enhanced severity of acute lung injury. We also had a closer look into the alveolar type 2 cells, because these are, of course, important for the surfactant system, but also then later on in regeneration. And we were interested in seeing if we have probably more senescence in these cell types. So this is a single cell sequencing data. When we isolated these cells, we could not see per se that we had a difference in senescence expression. When we looked here at P53, P21, also P16, in old type 2 cells versus young. But when we exposed them to LPS, we could see that these markers were increasing, particularly in the old type 2 cells with acute lung injury. Also for surfactant, like genes related to surfactant metabolism and to inflammation, we could see some differences. So here for the surfactant metabolism, we could see that with LPS exposure in the old animals, we could see this strong decrease in surfactant function and metabolism, but also an increase in inflammatory signaling in the type 2 cells. And notably though, in the old animals, we did see an increase also in some cytokines expression even without LPS. So we could see that we have these changes with acute lung injury, with the induction of senescence in type 2 cells and altered signaling. And these are all factors that could further actually accelerate acute lung injury. So we were interested if this is probably something that contributes to the injury, or if this is something that is a result of the injury. And therefore, we also used this Grelux-P mouse model, where we deleted turf in 82 cells, and we compared it to young animals without turf deletion and to aged animals, which were again like 18 months of age old. And here we used the lower dose because we didn't want to kill all the old animals again, but still we had a very poor survival rate for the old animals. The young animals, however, they all made it, no matter if we had a turf deletion or not. So we can see that the deletion of turf did not so much affect the outcome, the final outcome. Here you see the tissue of the turf knockout animals at day 7, and here of the regular young animals, and they both recovered after 7 days. And this was also reflected by the cytokines that we measured in the lung tissue here. But we could see, though, that the animals with TERP1 deletion in the type 2 cells, they actually had at day 4 a higher pro-inflammatory response that we could detect. So we can actually say that the induction of senescence further adds to the development of acute lung injury, but it is not like causing mortality as in old animals. So from that, we can kind of conclude that we do see pulmonary and systemic inflammation that is increasing with age. And this is also related to an enhanced severity and mortality that we see with acute lung injury. And that senescence does, in type 2 cells, does further promote the inflammation in acute lung injury, but it's not per se, or it was not per se acute, inducing inflammation in the lung in general, and also not enhancing the mortality. So we can also say that most likely there are other local and systemic components of inflammation that support the severity of ALI in old age. So in the next study, we wanted to address more the systemic component, and we wanted to have a closer look into the gut-lung axis. Microbial dysbiosis was also added as a new hallmark of aging, and this is like some changes that occur in the gut microbiota with advancing age. So we see changes in the composition, and we find, for instance, less diversity in the gut microbiota of the elderly. We see less beneficial bacteria that produce short-chain fatty acids. And short-chain fatty acids, they are known to support the integrity of the epithelia and also to have anti-inflammatory properties. So the elderly also show actually a more leaky epithelial barrier, which also probably allows pathogens or LPS to enter the system and thereby also potentially promote inflammation. Now, in our study, we were interested here how this actually correlates if we have changes in the gut microbiome with aging, and is this related to inflammation in the lung or in serum. So we did collect fecal pellets and tissue and serum and performed the network analysis. And we also did provide short-chain fatty acids in the drinking water to young and old mice for 2 weeks, and then again induced lung injury with LPS and sacrificed the animals and compared the outcome. So here, this is the data that we have from the network analysis. So we have here in red positive correlation, like in violet negative correlations, and we see that some bacterial taxa, they actually do correlate positively with age versus others correlate negatively, and they also correlate with some cytokines we see in the lung, but also activation and numbers of some of the macrophages and monocytes that we see in the lung tissue. So we see this correlation, and when we did treat the animals with short-chain fatty acids, we did collect the lung tissue and we did proteomics analysis, and this is from ingenuity pathway analysis, that we see actually the difference in control animals that do not have acute lung injury induction. So we see here the short-chain fatty acid treatment in the old mice mostly reduced, everything in blue is going down, the response of phagocytes, immune cell response, endocytosis, phagocytosis, and activation of immune cells, which very much relates to what we see here. And in the young mice, we did not see much effects. And when we induced lung injury then and compared the response without short-chain fatty acid treatment and with short-chain fatty acid treatment, we could see here that we have much more pathways related also to immune function and everything that are enhanced in aging, and this response is then dampened with short-chain fatty acid treatment. So we can say that short-chain fatty acid actually improves inflammation and also in acute lung injury, and we see this correlation between the gut-lung axis that we found here. Now, in another study, we wanted to have a further look into now more a model of infection with Klebsiella pneumonia, and also address more the impact of the systemic component, like the hematopoietic system, and we did here bone marrow transplantation in old mice. So we did look into the inflammation in the lung, but also in the pleura, because Klebsiella pneumonia has also been reported in rodents to cause pleuritis and also in some other species. And here, again, we had a similar picture where we see like the controls, and here we have after 24 hours and 48 hours of infection, we see here in the pleura an increase in inflammatory cell recruitment in the old animals, which is not like present in the young animals, and also accelerated inflammation going on here in the old animals. And also here in these models, some of the old animals died during the course of infection, whereas all of the young animals did survive. Similarly, when we looked into the bulb cells and into the pleural fluid, we could see this increase in cell recruitment, and this was also reflected by the cell number. So we mostly had neutrophils that increased at 24 hours. And this was also the case in the pleural fluid, where we had this increase in neutrophil recruitment. So we could show as well in this model that we have more severe pleuritis and decreased survival in old age with an infection model. And as I said, to address the effect of the hematopoietic system, we did bone marrow transplantation, where we transplanted bone marrow from old mice or from young mice into the old mice that were previously irradiated. And after a recovery period, they were again infected with Klebsiella. And we could see that particularly in the blood, the colony-forming units of Klebsiella, they significantly reduced in those animals with the young transplanted bone marrow, and also the neutrophils in the pleura. And also here you can see that in the histology that some of the effects of the pleuritis were gone after transplantation with young bone marrow. And similarly, this is also reflected in the cytokines that were measured in the pleural fluid with the old bone marrow or with the young bone marrow. So we can say that the bone marrow transplantation and the hematopoietic system is also affected by aging, and the bone marrow from young mice actually did improve the course of infection in the old animals. Now, last but not least, we also had a closer look at models of ex vivo models with precision cut lung slices from young and old mice, as well as neutrophils as a main cell type that is being recruited through the course of infection. And we used infection with Pseudomonas aeruginosa with a laboratory strain, PA01, or with a clinical isolate that was at one point extracted from or isolated from a cystic fibrosis patient. And we looked at different time points of infections as well. So we looked at gene expression in microarrays with microarray in the PCLS and cytokine expression. And for the neutrophils, we addressed the net formation and cell activation in the neutrophils. And interestingly, only when we took the PCLS without extras of the hematopoietic system, we could actually show that we have increased in inflammatory signaling when we compare the PCLS between young and old. So this is the microarray data here, like with the pathway analysis with David. And this effect was also maintained with infection with both of the strains that we had here. So always the immune system response was more accelerated in the old versus the young. Also with cytokine measurement, this was confirmed. Here we did not see a difference in the controls per se, but it also has to be considered that this is very little tissue, and the ELISA is probably not as sensitive as a gene array here. So here as well, the PCLS from old mice showed more inflammation and an increased inflammation with infection. When it comes here to the neutrophils, it was slightly different. Here we had the net formation, so we did quantify the net formation with different means, but these are just two SEM pictures. So we can see that there was actually a difference in net formation between the two strains that we had here, but when we quantified it, there was no difference between young and old. However, when we had a closer look here at the activation of the neutrophils, for instance, CD11B or CD16 expression that related to migration or to phagocytosis, we did see this decline activation in the neutrophils from old mice isolated. And also with the cytokines, there was a tendency, not like at individual level in comparison, but within a group comparison, we could see that there's a tendency of a decreased response in the neutrophils from old animals. So we can say that the neutrophils are less activated, and this is also in comparison to the PCLS. When we did a co-culture experiment, we could see that actually the PCLS, the age of the PCLS, did derive or did promote the increased inflammatory response that we observed with infection, like for instance here. And the age of the neutrophils did not really add more to that. So that was another difference where we could see actually this increased inflammation that is occurring in the lung tissue, but like this decreased activation that we did see in bone marrow-derived neutrophils. So with that, from this study, we could, or these studies, we could kind of like show and conclude that we do see definitely pulmonary inflammation and senescence that is related to increased severity in early, like in the PCLS or regularly in the lung tissue. And however, inflammation in the lung tissue is not the only factor that aggravates acute lung injury, such as we did see also the hematopoietic system of young mice is much better actually in fighting actually the inflammation or also the infection and ameliorating acute lung injury. And also we did see when we looked at the gut-lung axis that reduction in inflammation in lung and serum can actually improve acute lung injury in old mice. So yeah, with that, we can say that actually inflammation is playing an important role also in acute diseases and acute lung diseases, and it certainly has several sources, but there is yeah, so and addressing and reducing inflammation in elderly population could certainly also be beneficial to a series of disease, acute as well as chronic disease. And I'd like to thank particularly the doctoral students from the previous lab where I was in Hanover Medical School, as well as here now, where I'm now in Berlin, and also my other collaborators that contributed to the study. And I'd like to thank you for your attention, and I'm looking forward to the discussion.
Thank you, Dr. Brandenberger, for a very nice talk. We are a few minutes over our allotted time, but feel free for the audience to sign off if you need to go, but I think we'll hang on for about 5 more minutes just to have a quick discussion about your talk from the panel.
Yeah, I can share the screen if you unshare the screen, please.
Sure, of course.
Thank you.
Well, thanks. Thank you for a great talk. I'm sorry I missed it, but your model there was IT LPS at the beginning, the first part of the talk?
Yeah, exactly. We first started with LPS as a model, just to look into the inflammatory response, and then later on we continued with bacterial infection like Klebsiella or Pseudomonas.
So, you know, when I think about at least LPS, I think, you know, and I think a lot about inflammasome activation, and your data at the end there in the PCLS also looked at, you had the biggest signal in IL-1 beta, I think. You know, do you have any idea about what might be lowering the threshold for inflammasome activation?
Actually, yeah, we also did, I did leave out that one, but also we did some experiments in the Klebsiella study where we used caspase-1 inhibitor, and we were actually able to dampen the inflammation with that. So this seems to be also a promising strategy towards that end, but we did not go into long run experiments to see. So we did like this short term, like 24 hours experiment. So if that would help in the long run for the survival, this still needs to be looked at. But I think IL-1 beta would be a promising candidate. I agree on that. Yeah.
That's great. Thank you so much for Christina. Wonderful presentation. And also the wonderful by Dr. Rao. So thank you again, both of you. So me, Irfan Rahman, I have a concluding slide here. And again, I would like to thank to our co-chairs, Beata and Mareka and Ben, for hosting these things. So this is a really good, exciting time for us. So lung aging session in the ATS is proposed with our colleague from the NIAA, National Institute of Aging, Dr. Yao. And please look, the program is Monday 20th at 12 noon. So that's what we arranged. So hopefully you guys will join us in San Diego. So the membership, we have a lot of different things. We are making subgroups. So please do join us for your potential role and ideas, interactions, and any feedback would be welcome. This is for you. This is for the member. This is, we cannot get success without your efforts and your willingness to cooperate and put forward the helpful suggestions to us. So the ATS session, Sunrise, Meet the Professor, and all those new events will be out soon. So please try to submit for 2025. And that would be lung aging. And we don't know the theme of the next year's session, but this year is omics. So please try to attend. So ATS awards, we will be helping you if you'd like to submit anything for award, research, any RCMB activities. Please check out the RCMB websites and our come to the ATS on the evening reception of the RCMB. So with this, I would like to give my microphone to my colleague, Dr. Ben Singer, and also other colleagues here. And thank you again for joining us. So Ben, please go ahead for a minute.
Yeah, great. Thanks, Irfan. So going forward, these webinars are really an important part of what the group does here in the Aging RCMB group. And reach out to us if you want to host one or plan one. We have a lot of different ideas within different domains of aging biology, including focusing on multi-omic studies, looking at interventional studies to try to understand preclinical interventions for translation, other models of aging. We heard about PCLS today, organoids. There are many different ways to study aging in a model system. So these are all great fodder for webinars. So thank you again for joining and for sticking with us post time. But I hope you got a lot out of this webinar. I know that I did. Thank you to the ATS staff, Bridget in particular, and to our speakers and panelists. Have a great afternoon, everyone.